Hello everyone, welcome back to the Genomics Bootcamp and to the Introduction to Genomics lecture series. This time we continue with the presentation on the genomic admixture. Before we dive into the subject, there is a bit of a refreshment from the previous knowledge and also some information that might be useful in understanding of the topics that we will be speaking about. So we continue to speak about SNP marker data that could be used in genomics in many ways to characterize entire populations or individuals. This could be achieved even without the necessity of traditional pedigree data. Of course, we talk about traditional inheritance mechanisms in diploid organisms when we look at the inheritance mechanisms on a chromosomal basis, and in here, the so-called recombinations are also playing a major role, mixing up parental haplotypes and creating new variation in the population. The number and the size of these haplotypes is influenced, among other things, also by linkage disequilibrium or LD that characterizes the degree of relationship within these chromosome segments. When we talk about admixture, we talk about situations when individuals from two or more previously separated populations begin interbreeding. And after that, their offspring is kind of a mixture of these two parental populations. Now, the center of this picture is such an admixed individual. And if you are familiar somewhat with livestock breeds, then you probably guess that this is an admix of the Holstein cattle because of the very typical patterns and shapes which this individual has. But also, if you know the Holstein breed, so you also see that something is really off because some of the traits of this individual or some of these features are absolutely not typical to this breed. And this is because it is likely an offspring of a Holstein individual and an other breed. So this is how a purebred Holstein cow looks like, and the other breed is the Ancole cattle, which is a very particular and beautiful cattle breed from Africa. You see that the extreme horns are one of the dominant features of this breed, and the individual you have seen before basically carries the external characteristics of the Holstein and the Ancole cattle at the same time. Now, the admixture events or crossbreeding, especially in livestock, is not out of the ordinary. It's used in many times and many ways to improve populations or just basically creating new hybrids. Of course, it could be used in all other species as well, including plants and humans and everything. But in this particular case, I focus on livestock because, well, this is a bit closer to my field of expertise. So the picture, what we see here is a scheme for a particular admixture event where we have two breeds, two purebred breeds that are mated, and then the offspring is basically has the proportion from breed 1 and breed 2. Well, in this case, it's breed 1 and breed 0. The decimal numbers that you see in some of these shapes are basically the proportion of breed 1. And if we have just the pedigree information, then we work under an assumption that exactly half of the genome comes from one parent and exactly half of the genome comes from the other parent, which is of course true. And then if we have a so-called F1 population, when we have one purebred individual and other purebred individual, then the proportion, breed proportion for breed 1 in this case is 0.5. If we would mate this individual with another individual from breed 1, so basically we have a proportion for their offspring 0.75 from breed 1 because let's just half comes from here and half of the half comes from here, so 0.25 from here and 0.5 from here. So that's why this is a 0.75 for the proportion of the breed 1, or in other words, 75% of the genome of this individual is from breed 1. Of course, you can have various combinations, and at some point You can interbreed also two crossbreds, which also, well, result in a different crossbred with certain proportion of breed 1 and breed 2. So basically, this is how the breed proportions are calculated based on pedigree information, which such a scheme does not take into the account, however, are the so-called recombination events. And we know that such events are happening on every chromosome. So basically these numbers, especially when we are getting to more complicated crossbreds or more complicated admixture events, these numbers might be off from the expectations. Of course, another situation might be that such pedigree relationships are not available at all, which is also many times the case. So we have absolutely no idea which breeds have contributed as immediate parental generations or higher up in the pedigree. A solution is to analyze the admixture results based on purely genotype data. So what we have is we genotype these individuals with the SNP chips, and we use this information along with SNP chip information from any purebreds to determine the breed proportions in all individuals. The admixture proportions are, of course, shown in numbers, could be either decimals or percentages, but they are also very often visualized in so-called admixture plots or admixture graphs. I will show you such admixture graphs in a moment, but first I want to take a bit of time to explain how to interpret these admixture graphs. So basically for each individual, usually you have such a bar plot or basically a stripe, which is of different colors, depending on how many populations we consider in our admixture analysis. The entire length of the bar is, well, the 100%, and then the proportion of these colored bars are basically the proportion of those populations for that particular individual. So in this case, let's say that we go for 3 populations. So this individual is an admixed one from 3 populations that contributed to its genome. And it is about, let's say, 60% from population 1, about 25% from population 2, and about 15% from population 3. So for these numbers, I was just kind of guessing based on the lengths of the bars. And in reality, of course, in your results, you have exact numbers, and these exact numbers are visualized. So here is an admixture plot for actually 4 populations. So these are 4 sheep populations from Ethiopia. And you could see that basically they are quite different when it comes to admixture proportions. So this would be population 1, 2, 3, and 4. But what you can also see that some animals are kind of strange within these populations. Let's say in this population, so there is this one individual that for some reason is really similar to those that are from this other population. So it could be that basically there was a transfer of individuals, or this particular individual transferred from this region to this one, or it could be, you know, mislabeling or anything like that. So this is just to show that even without pedigree information, we can actually see the admixture proportions of individuals, and then also we can draw conclusions out of it. Of course, if we see strange cases like this, then this is a prompt for us to investigate what happened. Also, what we could see is that within these populations, sometimes there are some really specific individuals which seem to be interesting in some way. So you see that this analysis was done with 3 parental populations in mind, and many of these individuals are actually the admixed forms from these 3 parental populations. But in some cases, you see some of these bars are just with a single color, indicating that these could be somewhat different from the typical individuals in this region. So, for example, these are these guys here, or also in the other populations, you see other animals that are of a specific color. meaning that according to the admixture analysis, they consist only from this one parental population. Well, there is a specific set of questions to determine how many parental populations there should be considered. And of course, if we increase the number of parental populations, that could lead to, well, more exact answers, or on the other hand, it could also lead to even more confusions within the individuals and within the pictures of the admixture proportions within populations. I will not go into this question right now, but this is the so-called K value in the admixture analysis. And basically there is a question that how many subpopulations or how many parental populations should be considered for a particular admixture analysis. Also here I would add that the parental populations are not necessarily need to be separate breeds, but it could be various subpopulations within a breed, for example. This is a PCA plot for exactly the same animals as you seen before in the admixture plot. And here I just wanted to highlight that even though these populations are very distinct in a PCA plot, still they seem to be very interesting when it comes to a more detailed admixture analysis. Generally speaking, the admixture results in the introduction of new genetic lineages into a population and basically could occur when a geographic barrier separating two populations disappears for some reason. So these could be some natural events, but also, of course, anthropogenic, meaning that they are the results of human interventions. This is very often the case when it comes to creating crossbred populations in livestock, or of course also in plant breeding and elsewhere where human interventions are relevant. As a follow-up, I would like also to give another example of a crossbred population that was created by human intervention, and this is this so-called Swiss Fleckvieh cattle. This breed was established about 40 years ago and basically crossing the Fleckvieh or Simmental cattle with red Holstein Friesian. So pictures for the two purebred populations are here, and basically the goal was to create a very specific admixed breed that has the advantages of both populations. And because this work is, well, relatively recent, we have also a wide range of crosses within this population. So we see here another admixture plot, well, similar to the previous one I showed you. So here are the two purebred populations, and this is entirely based on pedigree results. This is because I will then compare the very same individuals to the genomic admixture. So what we have here are the two purebred populations, the Simmental and the Fleckvieh, right now represented by numbers. And this part is the population that is crossbred in nature. So these are basically the admixed individuals. So we have individuals that are really high proportion from one breed, about 70 or 80%. And of course, the rest is from the other breed. And other side of the spectrum is that, let's say, there is about 10% from one breed and 80 or 90% from other breed. These individuals are ranked here. So you see kind of a decreasing line when it comes to admixture proportions. Also, you see in this picture is everything is really clean and according to expectations. Well, these are the very same individuals, but this time the admixture proportions were calculated based on SNP genotype data. And here you see that, well, nature is not that simple as the previous picture was showing to us. Now, I need to highlight or underline that the order of the admixed individuals here is exactly the same. So what you see here is generally kind of the same trend, that it's decreasing in terms of admixture proportions. But what you could also see is a considerable variation between the admixture proportions in the individuals. So for example, in this part of the graph previously, the individuals were quite similar in terms of admixture proportions. So I just switched back here. So it's, well, not very similar, but like kind of the similar in terms of admixture proportions. And here there are, well, quite a bit of differences ranging to plus minus, let's say 10% of admixture proportions for some of the individuals. The reason, of course, could be that there are some recombination events that are taken care of when we compute the admixture rates or admixture proportions based on genotype data. The other thing that you might notice here, that previously the quote purebred populations were of single color. In this case is not entirely true. And the reason for this, well, it could be, again, well, anyone's guess, but one explanation is that might be that some of the animals which were considered, quote, purebred are perhaps not as purebred, and they had some kind of admixture events in their pedigree or in their past. Or the other explanation also could be that some of the genomic regions are just very similar between the two breeds, which is then picked up by the software that analyzes the admixture proportions, and therefore we don't get such a clean-cut picture as if we would just consider the pedigree information. Also, here I would mention that, well, in this case, we are very lucky because we have both the pedigree information and the admixture information based on the genomics or SNP chips available. So both are analyzed for this population. Well, many times, as I mentioned, the pedigree information is not available. But what we see also from this comparison that generally the correlation between the pedigree and genomic admixture results is really high. So even if we don't have the pedigree information available, we could get really good results based on purely the genotype data. In this slide, there is a comparison of different types of approaches when it comes to correlation of genomic and pedigree admixture. And this genomic admixture was calculated by various means and approaches. So these are listed here. But you see that within the lines, these are pretty similar. Interestingly, if you decrease the number of SNPs, so this was after the quality control, about 40,000 remained. But if you just take the 50% of the SNPs or 25%, or you just go down, you still get quite reasonable results. So even with 1% of the SNPs, so if you just consider the 1% of the SNPs as before, you still get very high correlations in the range of 0.93 when it comes to pedigree admixture. For this picture, this is just a demonstration of the admixture results based on this 1% of the SNPs. You see here it's a bit more shaky, and well, basically we have a lot more variation as before, but in the admixed individuals, so you see basically the very same tendency when it comes to admixture proportions from the two breeds. So even 1% of the SNPs, also very low proportion of the SNPs, is able to distinguish between high and low admixture proportions. Of course, there is a question, is there a better way to do this than selecting random 1% of the SNPs? And the answer is yes. And this is the situation when you consider very specific SNPs, selected SNPs based on the so-called ancestry information. So what you can do to select these ancestry informative SNPs is that you compare the SNP frequencies between the purebreds. You could use specific approaches, for example, the fixation index or FST, to select the SNPs that are the most different between the two populations. And by using these SNPs, you do a much better job compared to a situation when you just randomly select some SNPs. because these SNPs, the ancestry informative SNPs, have the value that they are able to identify the populations based on their genotypes. So here is an example of such ancestry informative SNPs, or at least how they work, in this case from human populations. So I would demonstrate the idea behind the ancestry informative SNPs, showing the extreme corners of these graphs here. So here are a few SNPs that for a particular genotype, they are basically fixed in people with East Asian ancestry. So they are frequency is 1 or almost 1, but they are not occurring at all for people with European ancestry. On the other side, there are also SNPs that for certain genotypes are appearing really frequently, or basically they are fixed for European people, but they are basically non-occurring for people for East Asian ancestry. Of course, there is difference where you want to make this cutoff point, but basically you see that there is a large number of SNPs that are really characteristic for certain populations. And if you select these SNPs and you use specifically these SNPs in your admixture analysis, you can do a very good job to determine the proportions of certain populations, even with using really low number of SNPs. Back to our cattle study. So this is an example of such approach when we again look at the correlations with the pedigree admixture, but this time we select SNPs based on the FST value. So basically a higher information value, because the higher the value of the FST, the more distinct are these SNPs between the two populations. And you see that already with 5,000 or 6,000 SNPs out of the 40,000, if these SNPs are selected based on the FST value, we already do a really good job when it comes to correlations to pedigree admixture. And if we decrease these numbers, so basically increasing the FST value, that also decreases the number of SNPs here. So you see that we go up with the FST value, we go down with the SNP numbers that are considered for such analysis. But even with about 600 SNPs, well-selected SNPs, we are at a level of about 0.96 when it comes to correlations between pedigree and genomic admixture results. And even if we go super low with number of SNPs, about 50 SNPs, 50 well-selected ancestry informative SNPs give us about 0.9 when it comes to correlations to pedigree results, which is really, really great. The reason why is this so important is, of course, related to cost issues. So the SNP chips in general are really affordable, but well, they still cost money. And when the genotyping costs are an issue, and to be honest, the costs of anything are an issue always, then basically going for a smaller number of SNPs might help, because of course genotyping a lower number of SNPs costs also much less. And also, which is well, very important, is that well, here on this channel we speak about SNP markers that are coming from SNP chips, but of course these require certain infrastructure and processes behind them. But of course there are also other opportunities how to do SNP genotypes with other more accessible and more affordable machines. And those machines, for example, typically can genotype 96 SNPs. But these results actually show that if you have, let's say, two very specific populations in mind, you can select ancestry informative SNPs for them, and you can go as low as 96 SNPs that you might even be able to genotype in your own lab or the lab of your institute, and you still get a very good correlation when it comes to admixture proportions. So as I mentioned, selecting a smaller sets of SNPs is a possibility, but also what you could do is select a relatively low number of purebred individuals and still do a really good job when it comes to determining admixture proportions. So to continue the line of arguments that I was having before, so basically what is tried out here is comparing reference sets of different sizes. So in this case, what we will do is compare a situation when we have the admixed individuals and 100 individuals from both purebred populations. But then what we do is have just 50-50, 20-20, and 10 individuals from each of the ancestral populations, and we will see how well the admixture proportions behave. And what we see, surprisingly, that the number of purebred individuals or animals in the reference set had really almost no impact on the results. So even with 10 individuals, we are basically really good in terms of determining the admixture proportions when it comes to correlations with pedigree admixture. So in summary, for this presentation on admixture, I just want to highlight the main outcomes. So it is definitely possible to find out proportions of purebred individuals and purebred genotypes when it comes to crossbreeding and admixed individuals. These could be from two breeds, which is quite often the case, but the admixture analysis is also capable to determine breed proportions from more than two breeds. Even a relatively low number of SNPs is sufficient to determine the admixture proportions, and you can improve your chances and improve your results if you select these SNPs in a specific way, for example, based on FST statistics with the so-called Ancestry informative SNPs. What you also need most of the time is purebred populations, but actually you don't need too many purebred individuals to be able to determine the admixture proportions in your crossbred or admixed individuals. So I think we finish here for now. I definitely thank you for your time you spent on this video. If you liked the video, then just hit the like button. If you're not subscribed, I would very much appreciate if you do so. And for me, nothing remains just to wish you a very nice day.